guys, so I've got a different setup trying here. We're going to see if it works. Last time I told you guys I would kind of talk about some of these things that you're doing for your project. So I already talked about diatoms, dinoflagellates, and daphnia. Today I was going to kind of take a look at, in class, I posted today for you on Canvas that you could practice using microscopes on gizmos. In class today we actually looked at some of the organisms that you've been looking up. We actually looked at a few other ones too that might be worth adding to the list. But for now, I'm going to kind of show you a few of the ones that we looked at in class today, just so you can kind of see them, and I'll give you a little bit of information about them. So the first is going to be an amoeba, and the amoeba, which is spelled weird, amoeba, right? The amoeba is this guy right here, and it has a nucleus and it has organelles inside of it and stuff. But what's really cool about them, and the three that I'm talking about today specifically, Paramecium and Euglena will be the other ones. What's really cool about these guys is that they have what are called false feet. So all three of these don't have false feet, but have a way to move. So these guys are going to be zooplankton. They are microscopic, they're hard to see. In fact, you can't see any of these really with the naked eye. So when you see these, they're usually in freshwater. Some of these can cross over into saltwater, but for the most part, they are freshwater. Amoebas have a cell membrane that is very flexible, and it allows them to extend out these projections from their cells. So they don't always look exactly like what I've drawn here. These are little projections that can extend out. It's the cytoplasm moving outward, and then it will kind of stop there, and the rest of it will kind of catch up with it, right? So these allow it to kind of move along like a blob, right? And those are called pseudopods, where pseudo means false, pod means foot. So these are false feet. It allows them to walk, as it were, with feet. Now, if they're in water, they're really not walking necessarily. They are kind of swimming, but it's the same idea. And because they are so small, they're relatively neutrally buoyant, so they're not really going to sink, which allows them to stay in the water column and drift about and move in that way. So that's some information about those guys. They do also eat. They use a process that's called endocytosis, or I think it's phagocytosis, something like that. Anyway, they surround what it is that they're trying to eat with these pseudopods, and then they surround it and then kind of open up the cell membrane and then allow that in, and then they basically digest it at that point, right? So that also allows them to eat. So the pseudopod is for moving and eating, right? Sorry. So the next guy that we want to talk about is going to be the Paramecium. So the Paramecium, it looks like this. It's really, really small, obviously, but it has this kind of indentation in its cell wall. This one has a cell wall. It's a little bit more rigid, and then in that indentation, it allows water to come through, which filters it, and then it basically feeds by taking things in through that. Then all of these little projections around the outside are little hairs, and they can do this. And imagine if you're doing this, right, then that allows you to swim through the water column. So cilia then will actually help those dudes move, allows them to swim. They can actually move pretty quick. When you're looking at them under a microscope and they're alive, they'll actually swim past, and you'll be like, whoa, where'd it go? And you can't really catch up to them. So they are really fast, but they are single cellular, just like amoeba. They have a nucleus and another membrane-bound organelles, so they're eukaryotic. They are planktonic, typically in freshwater, and they are zooplankton because they can move around and eat. So that's paramecium, mostly in freshwater, and should be good there. Let's move again to Euglena. So this is our friend, the Euglena. This is actually the smallest of these three. Euglena is actually small enough that it could fit inside of a paramecium, and there would be room there. So he's pretty small. He's hard to see in a microscope. And what's even harder to see is his form of locomotion, or his form of movement, which is that tail right there. That tail is called a flagellum. You might see the word flagella. That's just the plural form of flagellum. You should have already seen this in dinoflagellates. Dinoflagellates have a flagellum as well, and that is a whip-like tail for swimming. So they can swim with that and move with that, and they can forage for food with that. I believe some Euglena are also photosynthetic, so some of them, that ability to swim then allows them to swim towards light. I'm not sure if they are do respiration or have to get food, so you might look that up yourself. But that is your Euglena. Again, it is a single-celled organism. It's got a nucleus inside of it and then membrane-bound organelles as well. But the key is it has that flagellum that allows it to swim. Other than that, those are the three that I was going to talk about today. And then we looked them up here in class. While I'm here, I'm just going to go ahead and talk about a couple others. You're going to want to look up better pictures. You're going to want to look up better pictures of these. Because I don't know that I do them justice, but I'm going to go ahead and talk about them real quick. The first is foraminifera. So foraminifera are marine, and they look like these really ornate Christmas tree ornaments, like you've made some glass Christmas tree ornaments, and they look really cool. And so they have this kind of cool look. They have lots of holes on the outside of them, and then they have a lot of them will have different spiky appendages coming off of them. So on here I've put holes and spikes kind of on my drawing, and my drawings are bad. Go look some up. They're kind of cool looking. But anyway, they have spikes. These spikes will actually slow down their sinking rate. And then also, since they're spiky, kind of like a cactus, right, that can help them not get eaten by other larger things, possibly. So obviously you don't want to get spines stuck in your throat. It makes it harder to eat. So spines can be slow down sinking rates by basically having, arms stretched out. If you're sinking in the water, that adds more surface area, which then adds to your ability to not sink as fast. So those help with two things: sinking as fast, and then they're kind of a defense, right? Defense mechanism. And then they're mostly, I think they're usually silicone-based outside shells, and then inside they have like a soft gelatinous body, so kind of like a cytoplasm type thing. And what they can do is outside of those holes, they can extend part of their cytoplasm, kind of like a pseudopod, right? And they can actually grab prey and pull it back in. So that's kind of cool. They're relatively interesting. And then I'll let you find the rest. I think they do binary fission. Their reproduction, but I can't remember. I haven't looked at it in a while. Then Radiolaria. These dudes look like snails, right? And so they actually have a hard shell, and then they'll also have a soft internal body, and that allows them to, have projections that come out, and then they can eat things or move kind of like a pseudopod as well. So those guys are very similar to each other. And then some of them are a little different and do big differently. Some of them like that, but I'll let you look those guys up. I would like you to add a Volvox. I forgot to put Volvox on there. So add Volvox just because it fits well with all of these. But basically a Volvox looks like a big beach ball. And you can actually see these with the naked eye. In fact, let me see if I can pull up a slide that we looked at today. And maybe you can see through the camera, you know, with your naked eye where the Volvoxes are. Now this is going to be a cluster of Volvoxes, but with your naked eye, you can actually see. Let's see if we can get that to work. So you can see there the blotchy purpley stuff on there is a Volvox, and then let me see if, just out of curiosity, if I can get that to show up on a microscope for you. Alright, so I'm going to hold this up to that and see if I can get it to work here without messing with my You can see them there. Kind of hard because it's so bright. Hard to hold steady. So I'm gonna let that be it. And so as I was watching the film, I guess that I made, I was talking about a Volvox, and I absolutely misspoke. You know, I got to thinking about it, and I was like, I don't think they're a single-celled organism. And so I looked it up, and sure enough, I was wrong, which I know never happens. Actually happens quite a bit, but anyway, so I wanted to just kind of jump on here and set the record straight. Here's the Volvox. The ones you saw on the microscope were actually kind of a purplish color. That's just because they're stained that color. If you see them in real life, they're green. Kind of cool. Anyway, they're colonies of cells. They're not a single cell. So some colonies, it says, can be up to 50,000 cells. That's a lot of cells. And so they basically are colonizing together and working together to basically create you know, something that kind of work for the common good, as it were. So read about them a little bit. They're kind of cool, and add them into your project. If I don't see them in there, I'm not going to completely hold it against you, but I might ask you about a Volvox. So add them in there, and get them added to your list. But yeah, they are mostly freshwater, and yes, are really big. You can see them with your naked eye, which makes sense that they would then be colonies of cells and not a single cell like my dumb self said. So when you see that drawing on the video that says there's a nucleus, there's not. There's just going to be like 50,000 nuclei because there's a bunch of cells in that colony. So ignore me, but then come back and you're going to talk about some other stuff. They're really cool when you see them living. They're green, and they just kind of float through like a big old beach ball would be floating through on the slide, so they're kind of cool. Then let me come over here. The other one we looked at today was a Vorticella, and that is one of the ones that was on your sheet. But a Vorticella is really cool. I'm kind of a nerd when I say it's really cool. It's cool to see living. Like, we looked at them in a microscope and they're dead, they're already glued to the slide, that kind of thing. But you can, if you actually look close enough, you can see two different kinds sometimes. Let me see if I can blow this up for you. So this would be a Vorticella right here. And usually what happens is if you have like debris, so like sticks or gravel or anything that's in a pond, these guys are freshwater as well. What they do is they have a stalk, right? And that stalk basically is kind of like a holdfast. So they stick to substrate, and then that stalk is long, and then their cell body is this bit here. So it looks like a tuba, kind of. When you look at them under the microscope, I always call them the Pac-Man ghosts, because that's what they look like. But they have a string that they're tethered to some sort of substrate, and they basically can tighten up or coil up that string, that stalk, and make like a little spring, so that if something is coming along that they feel like they're threatened, then they can pull that tight, and that pulls them in closer to the piece of debris that they're hanging onto. Or they can also relax it and then come out, and then they actually will do filter feeding. So you know, if there's particles and stuff, they can actually move out and come back, and move out and come back, which allows them to essentially hungry hippo. If you've ever played that game, they come out and they grab food, and they come out and they grab food, right? And that's kind of the same idea here. So Vorticella are kind of cool. The last one here is Hydra. And Hydra are another big one. I'll show you a slide of a Hydra real quick. So you can see that they're visible to the naked eye. These guys are called freshwater jellyfish sometimes. Though they're not as big as a jellyfish. They are basically, let's see if we can do that again. So you can see that red kind of stain there on it. That is the Hydra. And if I put a Hydra on the slide and try to show you again here, they look like a I think there's a comic book guy that's a Hydra, right? That has a bunch of legs and stuff. Same thing here. These guys have tentacles, and if we wanted to see them, it would look like this guy here. Let's see if I can get this to work again. But basically, kind of hard because there's so much light coming. Second you got to see it, that maybe good as it gets, but you can see at the sorry, you can see at the top there are tentacles, and then there's a body that comes down and away, and you can pause that glimpse that we got so you can see it. But basically I also have it there on the board. It has tentacles, and those tentacles do have the ability to sting, but the sting is obviously that thing is microscopic, so if it were on you, you wouldn't even really be able to see it, but you might feel a little, tingle from it. If you were in a pool of them, you might feel like your skin is tingling. So they do have the ability to sting. They do capture their prey. Again, they're called freshwater jellyfish sometimes. But look those guys up too. So Hydra and Volvox. So add those to your project there. That way you can look at those guys, and I think I've talked about most of them now. So I'm gonna go ahead and end this here, and if there's anything I need to add to it, I'll add another video down the road.